Good morning, students. And today I'm going to discuss about the development of embryo sac and the types of embryo sac. See, before that we are discussing about female gametophyte and embryo sac structure also, and also we are discussing about oval structure and mm, mm, oval types also. See, we have in angiosperms mainly six different type of embryo ovals are present. Now I can I can explain the structure of the embryo sac. That is nothing but this is the structure of embryo sac. Majorly the embryo sac first of all discovered by Strasburger scientist in the plant of Polygonum. Now the embryo sac structure we can see mainly three regions, that is antipodal and also axil and polar nucleus in the central. See the antipodal, which is located in the ovule towards the chalazal side, and axil, which is located to the micropylar region. Why? Because the male gamete, either pollen grain, the pollen tube enter into the ovule through micropylar. Then the pollen tube, which are having two male gametes, one male gamete fusion with the axil, then the male gamete the pollen tube enter into the, most probable we have three different type of pollen tube enter into the ovule, but the chalagamy and the mesogamy and porogamy. See, most of the pollen tubes are enter into the micropylar region only. See, and this is the axil and synergy cells, which is located towards the micropylar region, and the antipodal cells are also having haploid cells which is not participating in the vegetative reproduction. Then in the central site, that is the polar nucleus, which contain two set of two set of the chromosomes, which is nothing but diploid cells, that is the secondary nucleus. And also the axil also having only haploid cell, and the synergy cells are also haploid cells having only one set of chromosome. This is not participating in the reproduction, only axil will participate it, and secondary nucleus is also participating in the second fertilization process in the triple fusion. This is all belongs to before the class, I think so. Today I'm going to discuss about the development of female gametophyte, that is nothing but polygonum type. See, first of all, megasporogenesis. What is the condition of megasporogenesis? Yesterday I am discussing the what is the megasporogenesis. When, whatever and whenever, even fertilization time and normal time, the mother cell condition is always diploid in nature. See, here they are telling megaspore mother cell. See, megaspore mother cell is also having diploid cell. We can mention in the diploid cell, the diploid cell which is participating in the meiosis division, which is producing four cells, that is four megaspores or four tetramegaspores. These four megaspores are haploid. See, in the four megaspores, the three are degenerated, only one is functionalized. This functionalized one megaspore is participated on the further division, either further development of polygonum type. Then and this is the common development of the are four tetramegaspores. In the three are degenerated, only one is functionalized. This is the one functionalized megaspore, and this one functionalized megaspore is participated mitosis division. Here very important, meiosis and mitosis. What is difference between meiosis and mitosis? Nothing but the meiosis division, but the mother cell which are having two set of chromosome and which is participating meiosis division, which producing four cells, those are all are haploid. Then mitosis division is nothing but whatever condition haploid and diploid and triploid mother cell which producing only two cells. The functional megaspores is here participated only one mitosis division. Just wait. The functional megaspore is participated one mitosis which producing two cells. See, these are the two cells. These two cells are which participating one more mitotic division which producing four cells. Two which located towards the chalachal side, and remaining two which is located to the micropylar region. Okay, then the four cells are again which participate in one more mitotic division which producing four cells. Then each region which are located four cells, then the chalazal four and remaining micropylar region also four. Then after that the formation of eight cells in the cell, then three are which is modified, which is form the three nucleus which is form a cell wall which is converted into antipodal cells, and remaining three towards the micropylar side which is form a cell wall which is formed egg cell and synergy cells, and one from each region migrated into central side which fuse, which is form a polar cells. The polar cell which contain two set of chromosome. This is the common EMBRYOSAC development, and this is first discovered by um, Strasburger scientist in the plant of Polygonum. This type of EMBRYOSAC is called as Polygonum type. Next, and we can observe embryo sac types. So what is embryo sac type? Actually in angiosperms we have only one type of embryo sac type is present, that is nothing but 8 celled and 7 celled and 8 nucleus embryo sac is present. And here different type of megaspore types are present in angiosperms, that is monosporic type and disporic type and tetrasporic types. See, monosporic type. See, what is monosporic type? This type of embryos are development only one megaspore will participate. Only one megaspore is participated. Monosporic, which means which is participating only one megaspore is participating, and remaining three are degenerated. And disporic, in this type of embryosac type which is participating two megaspores, I mean remaining two are degenerated. Tetrasporic, after that I can explain. Today I am explaining only in detail the only monosporic type and disporic types. See, the monosporic types again classified into mainly two types, that is Polygonum type and Enothera type. See, the disporic is also which is having mainly two types, that is Allium type and Endermion type. These are the main monosporic and disporic types. Before that, and see, this is the common development of Polygonum type. The first one the one megaspore. This is the megaspore mother cell which is participating one meiosis division which producing four megaspores. These are four megaspores and three are degenerated. Only one is functionalized. Then one functionalized megaspore. Just wait. This is the actual Polygonum type. Why? Because and three are degenerated, one is functionalized, one functionalized which is participating meiosis 1 and meiosis 2, meiosis 3, and after that participation of the three mitotic division which producing 8 cells and 4 each pole, and in the each pole one cell, one nucleus is migrate into the central side, fuse together, which is form polar cell, and also remaining three to the upper side. These are modified as antipodal cells, the formation of the cell wall to the surround of nucleus. In the lower side also three is nucleus which is formed as new egg cell and synergid cell. This is the Polygonum type. This is the common embryo structure. Now I can explain Enothera type. See, this is the Enothera type where you can observe one megaspore mother cell. See, yes, one megaspore mother cell which is participating meiosis division which producing 4 megaspores. These are the 4 megaspores, and 3 are degenerated. Only one is functionalized. This is the functionalized megaspore. Then one functionalized megaspore which is participating only mitotic division, which producing 2 cells. See, and these 2 cells which is located mm, to the outside. Then the remaining 2 cells which is participating one more mitotic division, దాని సమవిభజన అంటున్నాం. ఇది సమవిభజన చెంది either participating 2 mitotic division which producing 4 cells. Then 3 are modified as egg cell and also to synergy cells, and in the lower side polar cell, this is applied cell. Why? Because the functionalized megaspore is applied, and here the polar is also applied. I can mention applied in nature. Okay, this is the Enothera cell. Next one I can explain diasporic type. See, what is the diasporic type is nothing but here also similar with before the development of the megaspore developing, that is one spore mother cell which is participating, one spore mother cell which condition diploid and which is participating mitosis, one mitosis division, not meiosis division, which producing 2 cells. See, one is degenerated, only one is functionalized. See, the micropylar region cell is degenerated, only chalazal region is functionalized. Okay, and one functionalized cell which is participating mitotic division which producing 2 cells. One is located to the outside, either chalazal region, one which is located to the micropylar region. We can observe in the central vacuole also. Okay, then these 2 are again participated in 2 meiosis divisions. See, you can mention as 2 meiosis 1 and mitotic 1 and mitotic 2, mitotic 2 which producing 8 cells entire cell. The 8 cells, only 3 which is located to the outside, these are modified as antipodal, and remaining 3 which is located to the chalazal region, actually the outside chalazal region and the lower set micropylar region. And in the central we can observe one from outside and one from lower side migrate in fuse, which is form a central cell, that is nothing but 2 set of cell, that is polar cell, that is having applied. Next I'll explain this is the common spore mother cell. The spore mother cell which is participating same the similar with ileum type. Endermion type is also having spore mother cell which is participating micropylar region and to lower side chalazal region, and here the micropylar region of the cell is degenerated and only one is functionalized, and the functionalized cell which is participating one mitotic division which producing 2 cells to the outside and inner side, that is these 2 cells are also participating 2 mitosis division which producing 8 cells entire. Then the 8 cells which is modified as complete embryo sac. See, this is the Endermion type. We can observe Endermion and ileum type, so very very important right now. Next one I can explain actually the tetrasporic type, the cells which means 1 place 1 place 1 place 1. Usually the tetrasporic, which is nothing but the megaspore in the megasporogenesis, the megaspore mother cell which is participating meiosis division which producing 4 megaspores. These are the called as megasporogenesis. These 4 cells which is participating all megaspores which is participating in the development of embryo sac is called as tetrasporic. See, the arrangement of megaspores are mainly 3 types, that is nothing but one, that is very easily the arrangement of the spores is 1 plus 1 plus 1 plus 1, which means, for example, this is the common embryo sac cell. One is chalajal and one is chalajal, and remaining one is micropellar and remaining to lateral side. The arrangement of the megaspores, which is located to the four sides out and inner in the lateral sides. Then remaining the second arrangement, that is belongs to 1. See the arrangement of the cells, two cells which is located to the outside and two cells which is located to the chalajal. This is the common. This is the second type. Then third one, the embryo sac which are arranged like this. Then mainly one cell which is located to the micropellar region and remaining three cells which are arranged in the micropellar region. These three main different type of arrangements are present. Okay, and now I can explain. Just I can tell the names of tetrasporic, not a cleared explanation. We have mainly in the tetrasporics mainly 7 different type of EMBRYOSAC types are present. That is, first one peperomia type, and second one pinia type, and third one that is plumbago type, and fourth one and edaxo type, and fifth one drosa type. This is a very important drosa type. Some one time the given in the MAC also. And next one, third one, sixth one plumbagilla type. See, we can see this type of different different type of the 7 different type of present. That is one more I can explain these 7. That is a peperomia type and pinia type and plumbago type and edaxo type, and also Pritillaria type and Plumbagilla type. These are the different type of EMBRYOSAC types which is present in the tetrasporic type. See, these are the common different type of EMBRYOSAC types we can observe. So very important for essay for tetrasporic. Even normal polygonum, generally in normal essay question they are asking describe the general structure of the EMBRYOSAC and give the development of EMBRYOSAC in common angiosperms. Sometimes they are asking given detail of polygonum type of EMBRYOSAC. See, what is polyembryogen? What is polyembryogen? Polygonum EMBRYOSAC type is nothing but which contain 7 celled and 8 nucleus EMBRYOSAC. That is a common EMBRYOSAC. Then after that, in this I can explain in these messy questions. First one, what is the condition of megaspore mother cell condition? Before that I can explain, that is 2n. Okay, in the meiosis division which producing how many daughter cells? Meiosis I am asking, only 4 daughter cells are produced. Then in the mitosis that quite opposite to meiosis, that is only 2. Okay, in the meiosis the 4 daughter cells, what is the condition of the 4 daughter cells? Is haploid. Why? Because the mother cell, this I mean in the meiosis the diploid should be the diploid cells only should be participated only diploid cell. There is no participated only haploid and triploid in the meiosis division. Then in the mitosis which are participated whatever haploid and diploid and triploid. See, the mother cell which contain 2 set which is modified into 4 haploid cells only. Why? Because in the meiosis we can observe meiosis 1 and meiosis 2. In the meiosis 1 which produce only the mother cell which produce 2 cells. Then the meiosis 2 the 2 mother cell which producing 4 cells. In the time of meiosis 2 the chromosome number is reduced to up to 50% of chromosome. If 50 the cell which are having 50 chromosomes, for example which is participating meiosis 2, then which producing only 2 cells, each cell which contain 25 chromosomes only, which means the reducing of chromosome number is up to 50%. Okay, in next one, what is the Hmm. Condition of secondary nucleus they're asking, that is diploid. Always the secondary nucleus is diploid. Some cases in the tetrasporic, some drusa and plumbagilla and pinia type, which we can see the secondary nucleus is condition is not 2n, which we can see up tetrad and hexad and octa and so on. So okay, next one. And next MSc question here. Who was proposed chalagogamy? That is true. Next, what is the definition of mesogamy? Which means the pollen tube enter into the ovule through integuments. That is very important. What is the condition of nucellus in the ovule? That is diploid. Next, which type of no, just wait, I'm asking one more question. In tetrasporic, how many megaspores are participated in the development of embryosac? 4. And next, what is diasporic condition? Which means which contain 2 megaspores which participate in development of embryosac. These are all the MSc questions you can observe. It's a very important from embryology. So many questions are asking from in the MSc. You can carefully study. If you have any doubts, you can comment in the comment box, and also you can call to me, students. Okay, thank you, students. Thank you, one and all.